So we'll have our next speaker, Noemi. She's going to talk about karyotype-driven adaptations linking nutrient deprivation and whole genome duplication to metastasis and stress tolerance.
Thank you for the introduction. I want to start with mentioning that the work Richard just presented was submitted in June 2024 to a journal. And as of 2 hours ago, we got an email that it was informally, provisionally accepted. So we were excited about that. I think it was one of the longest review processes that I've been witnessing. That was all just one round of revision. And I think it was in part, not to the most part, but in part because Richard was one of the most critical reviewers. He was reviewer number 3. And as a result, we have an approach that works really well for a difficult problem. So Alpha K, check it out when you get a chance if you want to learn about karyotype fitness landscapes. So we study aneuploid karyotypes over the past 5 years. Our aim was to contribute to a better understanding of how karyotype variation contributes to phenotype variation. Does this work? It does not work. Okay. So in the world of karyotypes, mutations are chromosome missegregations. So mutation, selection, drift are the 3 key ingredients of evolution. And so chromosome missegregations have been modeled mathematically for more than 2 decades now. And as a result, we have the ability to calculate mutation rate dependent karyotype transition probabilities. Here shown in this matrix, I'm just showing a very small part of this matrix. It has about 70 quintillion times 70 quintillion entries in reality. So it cannot be stored. It has to be empirically computed. And we contributed to derive the equations to do so. So we know how mutation works in this context. What about selection? Aneuploidy has been thought to contribute to drive tumor progression. It happens in about 88% of tumors are aneuploid. Close to 100% of metastatic tumors are aneuploid. They start being aneuploid early on, and they stay aneuploid throughout progression. And aneuploidy in normal cells is exceedingly rare. Unlike point mutations, even for the least disputed of all driver events, TP53, up to 30% of normal cells in certain tissue types harbor TP53 mutations, yet stay normal. Normal cells with aneuploid genomes, though, are very rare. And this is because aneuploidy is highly phenotypically penetrant. It's not that missegregations don't happen in normal cells, they do, but the daughter cells don't stay alive for very long. In addition, the same missegregation event that gave rise to this trisomy 1 daughter cell presumably also gave rise to a monosomy 1 daughter cell. So note that these 2 daughter cells are different from each other and from the parental cells. So there are 3 different genotypes we have here. In contrast, the sibling of the TP53 mutated cell is presumably identical to the parental cell. So in summary, the rate at which a population can diversify its phenotype via missegregation is presumably faster than via point mutations. And that's why it's thought to be a major mechanism for cells to adapt to new environments. And one instance where cells must adapt to new environments is when they colonize a new tissue. So we'll talk about this first, and then in the second part, I will talk about more gradual adaptations, changes in resources. So colonization is thought to be the most challenging and inefficient step of the metastatic cascade across experimental systems. And that's in part because when cells end up in a new tissue type, they must completely reprogram their metabolism in order to be able to thrive there. And there are approaches out there that can predict whether metastasis will occur, machine learning, deep learning approaches, but they cannot reliably predict when it will happen. And that's in part because the signals these approaches are based on, gene expression, they change over time. They don't stay where they are. So here are 2 papers that have computed metastatic signatures. Both deal with breast cancer as a primary. One predicts metastasis to the lung, the other metastasis to the brain. They've been around for a couple of decades as well. So we can take these approaches and essentially calculate for each transcriptome state what's the associated risk of lung and brain metastasis respectively. So we can do this for the primary tumor at detection, calculate the signatures and calculate what is the likely risk that this tumor metastasis will metastasize to the lung, for example. And the accuracy of this prediction, though, will decay over time. And that is because the primary tumor will keep evolving, and so will its transcriptome. So if we were to the tumor, say 300 days later, its transcriptome will likely have changed, and it may now feature a completely different risk profile than the initial measurement. So what if we could predict those transcriptome changes? And this brings me back to karyotype, because karyotype variation copy number alterations are the most dominant predictor of gene expression, contributing 85% of predictions in breast cancer. They can predict gene expression variants of 40% of genes. And as you have seen in Richard's talk, we know approximately what effect these copy number changes will have on gene expression. So with that, we can calculate expression-informed karyotype fitness landscapes. So what this means, if most of the variants that we see in this transcriptomes, between these transcriptome states, comes from karyotype variation, then our states are no longer floating around, but they're connected in a way that we understand. So in order to implement this, you actually don't need so many things. You only need X and A, the karyotype and the transcriptome, at the primary detection, and two operators, gamma and eta, that project the misregression event onto the gene expression state. And from there, you can calculate then the metastatic risk. In this case, fitness is metastatic risk to the brain and lung. And we did this for 13 breast cancer cell lines that you can see listed here. So for each of these, we obtained two karyotype fitness landscapes, one predicting risk of metastasis to the lung, the other predicting risk of metastasis to the brain. So here I'm showing two examples, one for Cal51, the other one for BT549. These are two breast cancer cell lines. On the left, we have the lung KFL, on the right, we have the brain KFL. And the XY plane is essentially a projection, a UMO projection of the 22 karyotype vector, and the Z-axis shows the predicted fitness. And this is work that has been started by Thomas Weiss, who was a graduate student and recently moved as a postdoc. And he has been since taken over by Tao Li in the lab. So essentially, these KFLs are maps of how selection will act on karyotype in the lung and brain, respectively. So we can calculate how we can use the statistical model that Richard introduced and the agent-based model and project how the population will evolve in the lung and brain microenvironment. And we can then ask, so here every trajectory is such a prediction, and it's color-coded by the breast cancer cell line. So we have two replicates for each cell line. And we can then ask whether these predict metastasis, and they do not in the brain. For the lung, however, we do see a correlation coefficient of 0.53 between the average slope of these curves and the metastatic penetrance to the lung. So this brings me to the second part. So far, we have focused on very sudden changes when cells end up in a new tissue, very sudden change in the microenvironment. We have done some long-term evolution experiments where we expose the cells to more gradual changes. But before I get into that, one other instance of a very sudden change is certainly when you take patient-derived tumor cells and grow them in a dish. And so we asked whether there are any systematic differences we see in the karyotype. And we do see dramatic differences in fluidity. Here shown along the x-axis is the total DNA content. And we see that across all cancer types studied, along the y-axis, in vitro ploidy exceeds ploidy from the primary tumor. So in no single of these cancer types do the primary tumors have a higher ploidy than the cell lines. And of course, there's a lot of things that differ between in vivo and in vitro settings, but one striking or obvious difference is oxygen. Atmospheric oxygen is 21%. Nowhere in the body do we have access to 21% oxygen. Oxygen level in the body ranges between 2 and 5%, and that's down to 1 order of magnitude less than in primary tumors. So we hypothesized essentially that low oxygen selects against high ploidy. And what also supports this hypothesis is a strong correlation between the oxygen level in normal tissues. On the y-axis, you see oxygen level in different tissue types in the physiologic condition, no cancer. And the x-axis shows the incidence of whole genome doubling in the respective cancer type. So therefore, we hypothesized that if we were to reduce oxygen level in vitro as well, we should also see a ploidy reduction. So we worked with this isogenic pair of SUP159, 2N, and 4N cells that Voral has also mentioned a couple of days ago. And Voral and Jackson exposed them to limiting oxygen levels over several months and took images at each passage to count cells and to compute their growth rate, which is what you see here along the y-axis for diploid and tetraploid cells. For each cell line, we have the control, which had access to a lot of oxygen, and the 2 test replicates, which had access to limiting oxygens. And I think I'm running low on time, so I'm going to speed up a bit. This monitoring of growth rate essentially also informed us when to prioritize lineages for profiling, in this case, karyotyping. And this gave us the longitudinal karyotype data that you see here. For each, we calculate the ploidy, and we do see indeed a ploidy reduction as cells grow under hypoxia. And there is no sign of saturation, so we predict if we were to continue these experiments, they will continue to lose ploidy. Surprisingly, the 2N cells, the diploid cells, had undergone a whole genome doubling that was positively selected for a few months after the experiment started. This apparently contradicting result, one explanation for this, the leading hypothesis at the moment, is that hypoxia, in addition to limiting energy, also increases misaggregation rate. It increases SYN and aneuploidy. And in that context, access to a tetraploid genome is advantageous because it can buffer the deleterious effects of chromosome misaggregations. And in line with that, we see that whole genome double population starts shedding off genomes, chromosomes after having undergone this event. So aligned with the pre-existing 4N cells, de novo 4N cells also depolymerize or undergo ploidy reduction. In summary, this images emerging where various types of stress, including hypoxia, nutrient stress, increases chromosomal instability that diversifies karyotype, and then selection acts on this karyotype and thereby produces effectively cells that experience less stress because they're better adapted under this condition. So this process over time leads to increased genomic stability. So we are essentially in the business of figuring out which environment will select for which karyotypes. And with that, I'd like to thank everyone involved in this project. Special thanks to Xiaoqing Yu, for the extensive bioinformatics analysis, and Anna Gomez and Andriy Marusik for sharing genius ideas and invaluable material and reagents and cell lines. And happy to address any questions you may have.